Hello and welcome to the Testing and Tracing for COVID-19 webinar. I'm Michelle Tapper from the Australian Academy of Science. And this is the second event in a series of 6 focusing on international scientific collaboration in the fight against the COVID-19 pandemic. Firstly, I would like to acknowledge the traditional owners of the land on which we meet today. For me, it's the Yagara people. I would also like to pay my respects to elders past and present. Now, at the start of the pandemic, there was huge demand from countries around the world to develop and acquire COVID-19 testing kits to identify infected people. Testing and tracing are among the key strategies for controlling and stopping the spread of COVID-19 around the world. And testing comes in a variety of forms. There are a multitude of tests to determine if you have the virus or if you have antibodies against the virus. And in many countries, there is widespread wastewater testing to identify the presence and prevalence of the virus in communities. All of these approaches are vital factors in determining the public health response in different countries, which includes lockdowns, border controls, and contact tracing. And joining me to discuss how these things have evolved during the pandemic is Dr. Paul Bertsch from Australia's CSIRO. He's the Science Director of the Land and Water Business Unit. He's also an expert in wastewater testing. From Taiwan, we have a Professor of Medicine from the National Taiwan University Hospital, Yee-Chun Chen, who is an expert in infectious diseases and vaccinology. We also have Professor Michael Bunce, the Chief Scientist of the Environmental Protection Authority in New Zealand. Michael is a DNA and genetics expert, and currently he's seconded into New Zealand's COVID response at the Ministry of Health. Thanks to you all for joining us for this very important discussion. And before we ask our panelists some questions and get the conversation started, we have some messages from New Zealand and Taiwan. Please welcome the Australian High Commissioner to New Zealand, Her Excellency, the Honourable Patricia Forsythe, AM.
A warm welcome from Wellington, New Zealand, to this webinar event on COVID-19 testing and tracing. I'm delighted that experts from Australia, New Zealand and Taiwan, all considered world-leading experts in their response to the pandemic, have been able to come together virtually to share lessons learned. Testing and contact tracing systems has been crucial to the effective responses to the pandemic and success in containing the virus. International scientific engagement over COVID-19 has been a key to growing understanding of the virus, and informing responses, and events like today help further that understanding. Indeed, in the big picture, COVID-19 has put science in the spotlight and underscored the importance of increasing science, technology, engineering and mathematics, STEM capability, a key priority for Australia. Australia has had a challenging year with COVID-19, as to New Zealand, but through hard work, we've each managed to avoid the worst impacts of the virus. I want to acknowledge my host country, New Zealand, on its developments of world-class contact tracing and testing regimes, a critical part of the highly successful COVID-19 response. I wish you well for today's event and hope that lessons shared today will help to strengthen responses to the pandemic and continue to build the linkages so critical to the ongoing success in fighting this pandemic.
Thank you, High Commissioner. Now please welcome Representative Jenny Bloomfield from the Australian office in Taipei.
Very pleased to join you today from Taipei, where I started this month as Australia's representative. It's great to see experts from Australia, Taiwan and New Zealand able to come together today through this webinar to share lessons on COVID testing and tracing. Australians had a difficult year in 2020, as did much of the world. But through hard work and determination, we now appear to have the virus under control. Taiwan has, of course, made global headlines with its own great success in managing the pandemic. And in many ways, this crisis has only deepened our cooperation. We are both doing our part to assist others in the region through access to PPE, medical equipment and vaccines. And we have a great deal that we can share in respect of our own approaches. Today's webinar will help further deepen these important exchanges. Thank you, and I wish you well with today's event.
Thank you, Ms. Bloomfield. We appreciate these messages and the positive collaboration between our countries to fight this pandemic. Now, let's start with you, Paul. Australia, like New Zealand and Taiwan, has been applauded internationally for the way health authorities have managed our COVID response. Can you tell us about the importance of personal testing and wastewater testing, and how has it helped authorities here to manage outbreaks?
Yes, thanks, Michelle. Well, obviously, personal testing, the clinical testing is the gold standard that's used, and it's absolutely critical in the contact tracing capability, and that'll be talked about by my colleagues on the panel in much more detail. In terms of wastewater testing is both complementary as well as unique. So it's complementary in that it provides information that you don't get from individual testing. So you're really aggregating up at the community level. And so you could begin, for example, to see outbreaks emerging at a fine level of resolution if you choose to go in that direction. And it's also unique in that it's providing early warning detection. And there's many groups around the world that have demonstrated that they get a signal in wastewater before individuals go forward for individual testing. And one of the reasons that is it's been estimated that between 40 to 60% of those that are infected are asymptomatic. That is, that they don't display symptoms. And typically individuals begin to shed when they are towards the beginning of infection. So even if they're pre-symptomatic, that is before showing symptoms, they typically would be shedding. And so the viral fragments that they're shedding in their feces would be showing up in wastewater. And here in Australia and Queensland, we actually demonstrated by going back and doing retrospective analysis back into February that we were able to detect positives up to 3 weeks before the first clinical positive actually was identified. So that early warning detection is really important, and it is quite unique. And as I mentioned earlier, the complementary part is instead of doing random testing on a large population, particularly when you have very few cases, you again are aggregating up through at the community level to get that signal.
So it's obviously a very effective tool, but some people might be surprised that wastewater is regularly tested to gain public health data. What sort of things is it normally tested for, and how easy was it to switch to COVID testing?
Well, Michelle, it's actually been used for biologics, but more typically, people think about the testing for illicit drugs as well as other pharmaceuticals. And so the testing for illicit drugs, again, gives health professionals information in terms of what particular illicit drugs are being used, in what frequency and what quantity, as well as when they're shifting in terms of what illicit drugs individuals are using. And again, it gives that community level resolution, which allows the health professionals then to go in and do more specific response in terms of their public health response at a community level. So it's actually quite powerful in that regard. Look, my colleagues at the University of Queensland, who we've been collaborating with, published a study in PNAS, the Proceedings of the National Academy of Sciences in 2019, where they actually went and sampled at the 2016 census and began to put data together for a whole host of chemical biomarkers, including diet. And they found very strong relationships between positive diets as well as lower use of things like alcohol, tobacco, and illicit drugs. And it correlated very well with socioeconomic as well as educational status. So this is being used more and more around the world. Now, in terms of the biologics, again, there's been certain countries that have been doing this for many decades. Israel is the classic one. Polio is still prevalent in the Middle East, and so they were looking for polio and other pathogens in their wastewater. And in 2013, they also had that early warning discovery of polio in their wastewater when they had no idea that it was in the community. And of course, the public health response then is to go out, making sure that everybody is vaccinated and really getting the word out that people need to be vaccinated because it is the virus is in the community and it's spreading in the community.
But how easy was it to switch to COVID testing? Was that a fairly simple process?
It's always complicated just because wastewater is a very complex matrix. So unlike many biologic fluids that humans are tested for, it is quite complex. But again, people have been measuring viruses, so it was really just pivoting to some new technologies. Now, one of the things that we've discovered, if you look globally, is that laboratories vary in their experience actually with looking at biologics. And so that's been one of the problems of sort of quality control, as well as understanding what the sensitivity of the technique is. So for those laboratories that have been doing this for a long time, it wasn't that large of a pivot. And then we'll talk about this later, I'm sure, but the international collaboration was just unprecedented from my perspective. I mean, scientists were coming together very quickly, sharing information and ideas. And there still is a collaborative that has hundreds of members globally that meets regularly and shares information.
Yeah, and we've seen that a lot during the pandemic. Scientific collaboration has been key to getting a lot of answers to many things. Is there a good example of how testing wastewater has helped authorities manage a public health response in Australia?
Yes, there is. So currently, we do pick up positives in. So 3 states are actually doing regular testing and posting it online, and that would be Victoria, New South Wales, and Queensland. Other states and territories are doing some testing, but not having real-time delivery of the information. And here in Queensland, we work very closely with Queensland Health. The Chief Health Officer has been using this, the positives that we pick up as a means to really raise the consciousness of the public, because complacency was clearly setting in because of the very low incidence here in Queensland. We've done extraordinarily well as a nation, we have, but Queensland particularly, along with a few other states and territories, and she uses it to again raise that awareness, really urge people to get out and get tested. They set up pop-up testing stations, and people do line up for it. And we can talk a bit about some of the positives for individuals that have recovered that continue to shed the virus that are likely not infectious. But that still is used in a way that's keeping the awareness up that people need to continue to practice good hygiene, social distance whenever possible, and really just keep their guard up, because we've seen how quickly these little clusters can get out of hand.
Yeah, we certainly saw that a couple of weeks ago in Melbourne. Now, Michael, New Zealand has pretty much eradicated COVID with the exception of a couple of small outbreaks. What do you think has been some of the key actions that have led to New Zealand's success?
Kia ora toto, everybody. Yes, thank you, Michelle. I think in New Zealand, we talk a lot about the team of 5 million, and that would actually really be one of the first points that I'd make, in that in this pandemic, sick people need to get tested, and we need a variety of testing modalities. Otherwise, we will not catch it and we will not get on top of it early. So New Zealand went very hard, and it went early with its lockdown, and people absorbed the social distancing, and they got tested when they got sick. On top of this, we had very, very clear messaging around the alert levels, and very clear science communication, and our elimination strategy ultimately was shown to be effective. But the key aspect of it was community buy-in to that, backed up by some really good science.
Yeah, I like how you say that it's a group effort with everyone that lives in New Zealand. But how did the testing and tracing influence that hard lockdown that was implemented early on in the pandemic?
So if we go back about a year from today, really, I guess COVID was just starting to get a foothold in the community. So in March through New Zealand, we had on average about 50 to 100 cases per day. But 8 weeks later, it was eliminated, and we had 100 days of absolutely no COVID detections across the country. And this elimination was possible because of the testing. Scaling up PCR testing, very sensitive molecular tests to test for viral genetic material. We were also scaling up the contact tracing and integrating the testing in with our contact tracing teams, and recruiting, different people from across New Zealand that have key skills, including epidemiologists. And we had really good community communication and buy-in from the community. In terms of the testing modalities that we're using, New Zealand has very much stuck to a line of using PCR, the polymerase chain reaction test, of photocopying up genetic material, if you like, and using the nasopharyngeal or nose swabs. And that still really is the gold standard. It is the test that can detect COVID the very earliest in a particular individual. And although there was lots of pressure around bringing in rapid tests, point of care tests, things like pregnancy tests for COVID essentially, New Zealand pushed back quite hard against this because we had very low community transmission, and it wasn't always just about getting quick and dirty tests. It was about getting accurate tests that could pick up cases very early so we could stamp it out very, very quickly. And ultimately, I think that was the best call to make.
And now, having almost eradicated COVID, or basically eradicated COVID, what sort of testing is in place to continue to monitor the situation in New Zealand?
Well, the nasopharyngeal swabs, the nose swabs, if you like, is still really much the gold standard, but things have very much matured over the last year, and how we apply those tests and when we apply them. So in our managed isolation and quarantine, we have day 3 and day 12 tests, and day 01 tests were brought in response to rising levels of COVID around the world. Border workers are getting tested weekly, again using those ultra-sensitive tests. But in high-risk sites, we've brought in saliva testing that sort of fit in between as additional tests when we need them. Sick people still get tested in the community, and that is by far the best way to try and see if we've got community transmission. ESR here, as Paul's already mentioned, are doing the wastewater testing, and that's moved from a research capacity into an operational work. We're also swabbing some environmental services to look at some outbreaks and where the virus might be detected. But overlaid on top of all that information is, two other forms of testing, which is genomics, which we can talk a bit more about, as also serology, and how they interplay, how you take this toolkit of approaches and apply the right tool for the right job, given the type of outbreak, I guess, that you might have, whether it's constrained at the border or whether it's escaped into the community.
So New Zealand is still being extremely vigilant, and obviously that is paying off. Now, Taiwan has also managed to keep the pandemic well under control, with less than 1,000 infections in a population of almost 24 million people since the start of the pandemic. Yee-Chun, how did Taiwan's extensive public health infrastructure help with testing and tracing early in the pandemic?
Michelle, thank you for having me to share experience from Taiwan. This question is a key for conducting the national policy that is targeting testing and contact tracing in Taiwan. It is very similar to New Zealand, Mike has mentioned, and then it is quite different from the mass screening in many other countries. Taiwan CDC, local public health, and the infection prevention and control in the healthcare setting have been strengthened after SARS epidemic. We built the partnership between different government sector and the healthcare setting. This partnership has established based on daily practice and reinforced by annual nationwide hospital audit by Taiwan CDC or local public health department. So we build on the daily practice, and this is the key to conduct very intensive case tracing in Taiwan.
Yeah, so a very intensive approach. And unlike Australia and New Zealand, Taiwan avoided a national lockdown. So that's pretty impressive. How did the country manage infections, and what systems were put in place, in addition to what you've already mentioned, to try and stop the spread of COVID-19?
Yeah, this is very important. We initiate containment strategy at the beginning. I need to emphasize that every year during the influenza season, government will announce and ask for the nationwide to strengthen the infection control and to prevent the influenza outbreak. So Taiwan effectively delay and contain community transmission by leveraging experience from SARS epidemic and the very prevent public awareness. And we integrate public health and the healthcare response network and the support from healthcare setting and industry, cross-department collaboration, and advanced information technology capacity. We have 3 tiers of control. We initiate border control at the beginning, and we have very clear policy to prevent the spread in the community and protect healthcare setting. This 3 tier is initiated timely from the very beginning. We also escalate surge capacity for the testing nationwide. So we have very comprehensive policy and implement very timely and adjust many times based on international or local epidemiology.
And it's been pretty effective because Taiwan has only recorded 9 COVID-related deaths since the pandemic began. Now, Paul, what was the role of international collaboration in making Australia, New Zealand, and Taiwan's testing and tracing responses so effective?
Well, Michelle, I mean, as I mentioned earlier, it was really critical. And I suspect many individuals know that our neighbors to the north, like Taiwan, but also Singapore, had really vast experience in this from the SARS-CoV-1 infection outbreak. And we were a bit shielded from that, and so we had a lot to learn from our neighbors to the north, for sure. And I suspect Michael can speak to how New Zealand used that type of information as well to build their systems. And then here in Australia, our previous chief scientist, Alan Finkel, started a rapid research information forum, which actually included some scientists from New Zealand and elsewhere in the region. And it was just, again, a way to share in real time, particularly early on in the emergence of the pandemic, of what best practices were. And then they eventually actually had a national study on contact tracing to where they went to every state and territory to really look at best practices and then make some recommendations back to the national cabinet, which has all the premiers from the states and territories involved, and they accepted those recommendations. And so that was tremendous. In the wastewater space, I've already mentioned very rapidly, there was a collaboration, a global collaboration, which now has a few hundred individuals or institutions participating. They keep track of the various things going on around the world. So right now we're aware of 49 different countries doing regular wastewater testing. There's over 1,350 online sites that are updated regularly with the data from that surveillance. And then there's also just continued information exchange. Although we do have a ways to go still in getting our methodologies worked out to be a bit more consistent in understanding the level of sensitivity. But it was unprecedented. I think everyone I've spoken to around the response, as you've seen with vaccines and other things, that the scientific community believed that this was an unprecedented, very rapid movement towards collaboration.
Yeah, definitely unprecedented, and obviously a pretty exciting time to be working in science, to have access to some of the greatest minds around the world. What are some of the challenges with testing wastewater here in Australia?
Well, as I mentioned, Michelle, there's differences across jurisdictions in terms of the various methods being used. And the thing about the wastewater testing is, again, unlike taking a nasopharyngeal swab, there's a lot of variability, potential variability that actually goes in wastewater sample. How do you sample it? Do you take a point in time, or do you integrate over time? And we've done studies demonstrating that you really do have to integrate over time to get the sensitivity up. But we know that there's all kinds of different methods, not only here in Australia, but across the world, that are actually being used. And then just how you process the samples. There was very little data actually on concentrating up these enveloped viruses. So we had to really step back and develop and evaluate methods. And a lot of individuals around the world, and again, that variation would be here in Australia, the actual ability to efficiently capture the virus ranges from 2% up to 67%. And the 67% is work that we actually generated to optimize that. And we're continually optimizing to try to get that up to a higher level and increase the sensitivity. So, you know, there are clearly a number of challenges that we're still working through. We're really hoping that we can get a network across the country to begin to work on standardizing. So they're doing interlab comparisons in terms of measurements around the virus, but that doesn't get to the very front end, which we've also determined is quite critical, as I mentioned before, how you sample. And I learned a lot from my Singaporean colleagues quite early on, because they did some very detailed work up pipe, as it's called, at the postcode level, but they were even doing dorm measurements on those dorms where those international workers lived, and where their second wave actually emerged from. And they have some really good data on how you need to best integrate the sample. And, you know, so they were integrating over a 5-hour period, but we typically integrate over a 24 to 36-hour period. But as I mentioned, others go and just take a grab sample, which is just one point in time, which is really problematic.
So obviously testing wastewater is very important for health authorities in managing a public health response, but what would it look like if we didn't test wastewater? Would we have more border controls, more lockdowns? What would the difference be?
Well, I mean, if you look at the early stages of the emergence of the virus, not only here in Australia but globally, there was just a lot of random testing going on. And I can give you an example. One of the first examples out of Massachusetts, there was a startup company that was partnering with Harvard and MIT, and they were measuring in a suburb of Boston. And at the time, there were 450 clinically detected cases, and they estimated that there was 5 to 10,000 individuals that were actually infected in the community, and that then did have a response of increasing that random sampling and also urging people to get tested. And it was clear that the underreporting was being picked up on wastewater, and that's been demonstrated more and more through big data analysis globally.
Thanks for that, Paul. Now, Michael, from your perspective, what was the role of international collaboration in making Australia, New Zealand and Taiwan's testing and tracing responses so effective?
As I guess Paul's already touched upon, I can't imagine a pandemic response that didn't involve a really strong collaborative threads that run through it. We simply just couldn't do what we wanted to do from a science response without freely sharing information. And New Zealand has benefited from that, as have many other countries around the globe. And to give you one example of that, Michelle, if we go back to the 11th of January, where Professor Eddie Holmes and his colleague Dr. Zhang from China published the first ever SARS-CoV-2 genome. So this is the base, really the 30,000 letter code that is the blueprint of the COVID virus, if you like. Now, that genetic code is absolutely pivotal in designing all the tests that we talk about, whether it's sewage tests or nasopharyngeal swab tests that many of the listeners would have experienced before. Those were developed just 1 to 2 weeks after that sequence was publicly shared. And after that, then those tests spread around the globe, and they have formed the basis of it. And I'd like to sort of maybe do a thought experiment. What happened if that data wasn't freely shared and we were months behind because there was delays associated with not sharing that information, and would be even in a worse situation than we have here. We know that the virus spreads really quickly and that testing is absolutely critical. So New Zealand, I guess, has learned a lot from its international collaborations, and even in the wastewater space, our wastewater testers at ESR have benefited a lot from the Colossus collaboration that Paul's a huge part of, and that's allowed us to roll out the methods here in New Zealand. So collaboration is absolutely central to everything to do with this pandemic response.
Yeah, there's no doubt about that. Now, you've talked about genomic testing. You're a geneticist. Can you just elaborate and explain a little bit more what exactly it is and how it is being used during the pandemic?
So yes, so when we test for COVID-19, when you get a test, when you go into a clinic and get a nose test, you're actually just photocopying up a small part of the virus to detect if it's there. And we can say, yes, it's there because we've detected a small bit of it. That is quite different from sequencing all 30,000 letters of it to determine what the genetic code is, if you like. So by sequencing the entire genome, we get lots of additional information. For instance, we could hypothesize that this looks very much still like a bat virus. So we've got information on its origins. We've produced the scaffolds we need to design all of those tests. But very, very quickly, it has gone past just saying, oh, where's it come from? And can we use this information to design other tests? Very, very quickly, it's been used in contact tracing applications and to monitor viral evolution in real time. And these are the variants that we've heard a lot about over the last 2 or so months. To give you a perspective on this, there's over 300,000 COVID genomes now being sequenced and publicly available, a huge resource. And it is absolutely pivotal that we keep an eye on how the virus is changing, because how can we, I guess, alter our vaccines or understand if we've got more transmissible or variant strains if we can't actually keep that surveillance up.
Wow, 300,000, that's an impressive number. What sort of challenges has New Zealand faced with testing and regulating the market?
Well, whenever you stand up these, I guess, tests in a very, very rapid manner, like every single jurisdiction is, there's always going to be some teething issues. So, across New Zealand, it was about standing it up at multiple sites, so we didn't have to fly swabs around the country to get them tested. And we've got 16 core sites now, so everything's within close reach. It's validating all of the tests. Across New Zealand, we're very interested in using sensitive approaches, and we want to detect a lot of swabs that we take especially around community outbreaks. Like at the moment, we want to detect them as early possible in the clinical course. And so that's about limits of detection, how sensitive of our tests. It's about throughput. It's about getting thousands of tests through and to be able to scale it very, very rapidly. And it's about supply chains as well, making sure we've got everything, all the reagents we need to do that. In terms of regulating the market, all the diagnostic tests for COVID in New Zealand occur inside if you say authorised, validated testing laboratories. New Zealand even put a ban in place to stop the import of COVID-19 testing, rapid testing tools, so people couldn't do it themselves. And that is because there's a couple of reasons. One is that we didn't want people sucking up reagents that were in short supply. But secondly, it was about the fidelity of the tests and that those were being spun into actually our public health response, which is really important.
So there are a lot of links to the chain and they all play an important role. How do you manage huge amounts of data like that and make sure that all those different components are working together?
It has been, and in some places, some of our databases have been showed wanting. So some of the genomic information, we've had to make new testing parameters, new testing databases to be able to store that information. So the interface between contact traces and genomics essentially had to be developed on the fly because we'd never done that before. We did have some health systems for normal sort of pathology testing, if you like, and they've stood up. But under pressure, when we're constantly reporting on a 12, 24-hour cycles, that information needs to be readily available, and repeat testing of people that have tested positive or negative and then positive again. We have to be able to track that.
Okay, that's important information. Now, Yee-Chun, I've asked the other two panelists, I'm going to ask you, what was the role from Taiwan's perspective of collaboration between other countries, between Australia and New Zealand, in making your testing and tracing responses so effective?
Yeah, international collaboration is very important. I very agree with Michael and Paul's comment. From the beginning, the information sharing between the government level and some virology data, PCR protocol, sequence data for tracking the source, all this is very important. In addition, I want to mention that because people across the border, they need to provide the test. So even the Taiwan people who are test positive in other country, the data will send back to inform Taiwan. They will conduct the source or contact tracing. So this kind of international collaboration is very important for public health within country and between country. Overall, all this information, either from virology or from public health, this information all facilitate to guide or modify our national policy. So this is very important. In addition to biotechnology or other development or research for vaccine, diagnosis, and therapeutic, the whole world work together is very important.
And Taiwan also has a network of public and private testing centres and labs. Has that enabled quicker responses and results or improved tracing at all?
In Taiwan, no matter public sector or private sector, all controlled by government, all the tests should report to Taiwan CDC through website. This website reporting system is established for many years for notifiable disease in Taiwan. So every test should report through this website, and every positive result, virology lab should report to government as soon as possible. And the government local public health department personnel will contact the healthcare hospital setting to initiate contact tracing as soon as possible and should be complete within 24 hours. So this is a nationwide mandated system. And all the budget is covered by the government, covered by Taiwan CDC. This is different from the national health insurance system. So it's a mandated. And because Taiwan announced COVID-19 as category 5 notifiable disease on January 15, and the physician should report to the government within 24 hours for individual fulfill the criteria for COVID-19.
So it's obviously a very effective system. Now, Paul, you touched on this earlier, but if you could clarify, that would be great. When you get positive COVID results in wastewater, does that mean people are infected in the community, or are they shedding the virus long after they've been infected?
Yeah, Michelle, we do know that they do shed the virus long after they've been infected. I mean, one of the reasons why feces is so common in terms of shedding is because there's a protein that's very common in the small intestine that we nickname ACE2, and it's known to have receptors that have very high binding affinity to this virus, and so the virus does concentrate in intestines. And so we now know we were getting positive in cans for some time, and we were thinking, because people do shed, so we of course we have people just passing through some of our areas that may have come from Victoria or New South Wales that could have been infected in the past, for example, and have recovered. But in this case, this individual had recovered and been released and went back home to Cairns. And 8 weeks after, we were still getting positives, but this individual then displayed symptoms again and went and got tested positive, and then tested negative. And so we generally suspect that that individual was continuing to shed up to 8 weeks prior post recovery. So that does happen. And as I mentioned earlier, our chief health officer still thinks it's a very valuable tool because, for example, when we first opened the borders, and this idea that maybe it was someone from Victoria, well, in some instances, could be that someone was infected in the community, and of course you want to have the proper response. And so again, just raising that awareness in the community. They had the pop-up station in Arley Beach. People were lined up to get tested, whereas nobody was getting essentially getting tested previously. So it was a very informative public health response that demonstrates again that to keep the awareness amongst the community that this virus is still with us and it can emerge at any time.
So it's a pretty sophisticated process, but how sensitive is sewage testing? For example, could you detect one case in a population of, say, 1 million people?
There are several reports from around the world where that they had strong evidence that they were getting about that, 1 in 1 million. There was a desktop study done at the Arizona State University, which indicated that under worst case scenario, and I won't talk about all the conditions that make it worst case or best case, but under worst case scenario it would be 1 in 100,000, and under the best case scenario it would be 1 in 2 million. Here in Brisbane, we know that we were detecting 1 in 300,000. So because we knew there was about 300,000 people in the catchment, and we knew at the time there was only one infected individual that had actually departed from the Ruby Princess and were in hospital.
That's so impressive to hear about. Now, moving on to you, Michael. Early in the pandemic, worldwide demand for COVID-19 tests had a little bit of a technical issue, so I'm just going to ask you that question again. I think we had a small computer glitch. Just talking about the demand for COVID-19 test kits. Worldwide demand far outstripped supply in the beginning of the pandemic. Can you just elaborate on that again for us, please?
Sure. Yeah, I was just saying that it was a real concern, the shortages, and we're still suffering those shortages today with pipette tips that are in short supply, to special types of tips to move liquids around that our robots use to run these thousands of tests. But a few things New Zealand put in place was to not all use the same platform, so we weren't after the same kits in every single one of our laboratories. We had contingencies in place to pool samples together if push comes to shove. That means, you might put 10 individuals from community tests into one tube and save reagents, do one test instead of 10. But the Ministry of Health worked incredibly hard in nailing down its supply chains to make sure the labs have got what they want. But I guess fundamentally, and this is where New Zealand has huge advantages along with Australia and Taiwan, is that if we keep cases low, we don't have to test as much. And that means that it puts less pressure on our laboratories and supply chains.
Thank you. And we've talked a lot about different tests, wastewater tests, personal tests, but what about antigen and antibody tests? How do they work and how do they help decision makers?
Yeah, we've talked about, genetic tests, as you say, and wastewater testing. So antigen testing doesn't test for viral genetic material, it tests for the proteins, if you like, on the surface of the virus. And it can do it quite quickly. And in some jurisdictions, it's been widely used, rapid tests. But it will miss some. It's just not as sensitive as the genetic tests that can pick up. As Paul mentioned, it's the fascinating thing about these PCR tests. Theoretically, you only need a couple of copies to start off, and then they kind of photocopy up, if you like. But that's not the same with antigen tests, which essentially require enough virus on a particular swab for it to light up and test positive. So if you've got full-blown COVID, some of those antigen tests are quite good now. But if you're just developing the infection, or if you're at the tail end of it, they're not as good. So that's the antigen testing. And New Zealand, again, has not applied antigen tests widely across its testing regime. In terms of antibodies, and for the listeners out there, when we mount an immune response to a virus or even a vaccine, we tend to generate these antibody molecules. And testing for antibodies through a test called serology is also really important, because we can determine if a person is just recovering from COVID, or if they might be in an acute infection, which means that they've just got it. So if someone is a historical case, like Paul mentioned in some of the wastewater testing, they will still have antibody response to this. So we can detect the antibodies in the system. But if they've just acquired it, we won't see those antibodies. And so we can determine if a test, if an individual is early on their infection path, or indeed if they're a historical case. And we're getting a lot of cases coming into New Zealand across our border, which we classify as historical cases, because there's trace amounts of viral RNA in the samples, and we have serology to actually back that up and say they've previously generated an antibody response. So we don't think they're infectious. We think that that's a historical infection.
Well, thanks for explaining that, Mike. Now moving to you, Yee-Chun. What is Taiwan currently doing to continue to contain the spread of COVID, keep businesses open and the economy operating?
We continue all the strategy which were initiated early during the pandemic, including the border control, early case finding, isolation, those sick, quarantine those at risk, and continue so-called new normal life. That would encourage people in the community, universal wearing mask, and emphasize the hand hygiene to prevent the transmission in the community. In addition, the government continue emphasize, reinforce the infection control prevention in the healthcare setting. All this is continue. And very important, all this should be done at a timely manner. As I emphasize that, the testing and tracing is initiated and highly integrated, and it should be complete within 24 hours. People just wonder, how can we do that? I can provide a data. In Taiwan, we test the number of people test per million population is only 7,000. This is quite different from many other countries. This is the reason why we are very focused, very targeting testing. So we can initiate the contact tracing during very limited manpower.
Thank you for that, Yee-Chun. Now we have some questions from the audience. They've been submitted while we're talking here tonight, and also they've been submitted before this webinar online. The first question tonight is for Michael. Lorraine Vanderlind asks, is it possible to test for COVID with a finger prick of blood?
Well, the amount of virus circulating in your blood is incredibly low because this is a respiratory disease. So finding actual traces of viral genetic material in blood is quite difficult. But again, these antibodies that I just discussed before kick around your blood. And so you can actually test for it indirectly, seeing if someone has generated an antibody response about it. We will see many, many more fingerprint tests in 2021 and 2022 as we look at these antibodies, because it will form the basis of what scientists call a correlative protection. And what that means is really, do you have enough antibodies that you're protected from if you get exposed to the virus? And so that's a buzz term that's going to really kick around over the next couple of years, correlative protection, and it's going to be taken from blood fingerprint tests.
Oh, thanks for explaining that. And obviously we're going to see a lot more advancements as time goes on, because things are moving at a rapid pace. We have a question for Paul from Agnes Boskowitz. When the virus is detected in sewage, is it then possible to do further testing and narrow down the source of the virus, maybe even to individual buildings?
Yeah, so Michelle, I've already mentioned dormitory level analysis. And in fact, in the US, when the students returned this past autumn, the dorms were being regularly monitored. And one of the early warning detection events was at the University of Arizona, where they actually got a positive in one of the dorms. They went in, they did individual testing of all 312 residents in those dorms, and only 2 were positive, and they were able to isolate them, and no other students were infected. So again, shows that early warning detection, because it's unlikely that they weren't showing symptoms at the time, so they likely would have not gone and been tested. And the other thing, of course, with college-aged, university-aged students is that many of them are asymptomatic anyway. So yes, you can go up to a single asset. We've done cruise ships, we've done long-haul airplanes, we've done prisons, and we've done aged care facilities, for example. And our colleagues in Singapore have been doing this from the very early days of the pandemic, and I suspect maybe in Taiwan as well.
Well, thanks for explaining that, Paul. Now there's a question for Yee-Chun from Dr. Salai Sin from the Burma Medical Association. They want to know, what would your advice be with regards to testing and contact tracing in resource-constrained settings?
Okay. Yeah, this is very important issue as a global issue. During pandemic, all the demand are increased enormously. So how can we conduct in a cost-containing manner? So this is why we initiate targeting testing instead of mass screening. This can contain the stuff we need for the lab testing. In addition, for every positive case, we initiate contact tracing. Then we can contain the risk before further transmission of the disease to more people. So the aggressive strategy initiate, implement timely is most cost-containing manner. The similar effort you initiate later on, the effect is lower and the cost is higher. So this is very important regarding the pandemic control and also applied to every infectious disease. And the testing is important for every emergent infectious disease because people are more familiar with diagnosis. Test is for diagnosis. Then we can initiate therapy. But for transmissible, highly transmissible disease, we need to consider the timely intervention to stop the transmission. Overall, it will be cost containing. So this is a very important issue because January 2020, we already recognized the shortage of surgical masks in our hospital, even in our hospital, because many stuff are imported from other country, and that country are heavily hit by COVID-19. So this issue is a important issue during globalization, no matter the stuff for testing or for treatment for others. So we need to have different thinking regarding the strategy, how to facing the pandemic or other disaster in the future.
Thank you for explaining that. Now we've just got a couple of more viewer questions. One for you, Michael. Can the tests determine if there's lots of virus or only trace amounts? And how was that information used?
The short answer is yes. The test can determine if there's not lots or not very much. We run these PCR tests which photocopy out bits of the virus, and they're typically, they're technically called quantitative PCR tests because they do actually tell you if you've got a little bit or a lot. Now that information can be sometimes misleading, but sometimes actually quite useful. So in that scenario before, I was talking about historical cases. Historical cases will only have very small amounts of virus traditionally. And so we can potentially look at ones that amplify up very late or have very small copy numbers and then look at their serology and look at that. The flip side is that whether people are in early on their infection, they typically have large viral loads, and we can actually sort of try and understand the peak of what their infection might be. Now, most recently, you would have heard about some of these variants, terms like B117. That's the variant first found in the UK. On average, the B117 swabs taken from people that have got B117 have between 2 and 16 times more viral load compared to the original Wuhan virus, if you like, the original wild type virus. And so the amount of viral load in there essentially goes on to thinking about, do those patients actually shed more virus? And is that the core of why they're more transmissible? So it's a piece of the puzzle. It's not exactly kind of one for one where we can read exactly how many copies you have, but we can get an indication if you've got a little bit or a lot. And indeed, and maybe Paul wants to speak to this with regard to wastewater. Overseas, they've been looking at the amount of viral RNA in wastewater and trying to mirror that with the pandemic curves, if you like, on a population level. And that's been working quite well as well.
Paul, did you want to add to that? Because I have a controversial question for you from Nancy Choi. She wants to know, which country do you think currently has the best tracing system?
I'll let Michael and Yee-Chun talk about that one. But in terms of the wastewater, look, Michael's absolutely correct. There are, as more data comes in, it gets the analysis begin to allow us to get better and better at estimating the number of individuals in a catchment that are infected. There is a range just because of the viral shedding does vary per individual. But as I mentioned early in the pandemic, it was clear that even with that wide range, there was gross underreporting going on in most jurisdictions, and the wastewater demonstrated that was the case.
Okay, thank you for explaining that. And just the last question is for you, Michael. Tracy Willis wants to know, how will we do it better next time?
Well, I think we'll be silly if we haven't learned an awful lot from this. We will continue to collaborate. We need to share. I think doing better next time involves actually some of the early surveillance work, and this is work that Eddie Holmes and his colleagues have long pushed around the globe, is that we need to understand the viruses that are in animals, because they're the viruses that become the next pandemic. So we can do better about understanding the viruses that are all around us. And we can learn from the lessons of how we quickly stand up testing capabilities, contact tracing, and real-time genomics. So I still think it's marvelous that 24 hours after we've got a sample, we can get an entire genome of a virus. 2 decades ago, that would have been a pipe dream. Someone said you couldn't do that. So we'd be in a lot worse situation now if this pandemic was happening in the year 2000, for instance.
Yeah, and just look at the progress that we've had in the space of 1 year, with many countries already getting vaccinated. The scientific collaboration and progress has been truly incredible. And this has been a very interesting chat with all of you. And that's all we have time for this evening. I'd like to thank everyone for joining us. And thank you to the Department of Industry, Science, Energy and Resources. And of course, a big thanks to our panelists, Dr. Paul Bertsch, Professor Michael Bunce, and Professor Yee-Chun Chen. Now, our next webinar in this series will be in March, and it will be about genome sequencing of COVID-19. Be sure to check out the Australian Academy of Sciences website for all the details. And don't forget to follow us on social media for more trustworthy and fascinating